really, if we measure high sensitivity C-reactive protein, that seems to be a very good general marker of systemic inflammation. And systemic inflammation will contribute to neuroinflammation. Whenever you have inflammation in your body, it basically will loosen up and alter the permeability of your gut. So the tight junctions in your gut. And also we have the same tight junctions on the blood-brain barrier. So these tight junctions are very sensitive to the level of certain cytokines that are associated with autoimmune diseases and systemic inflammation. So we can kind of make the blanket statement, whenever we have inflammation in our body, in our joints, in our gut, that inflammation will influence the permeability of the blood-brain barrier, and that will contribute to neuroinflammation.
Well, good. Thanks for doing this again. Appreciate it.
Yeah, my pleasure. Great to connect with your community.
Yeah, and so everybody's, like just as per usual, these guys may type in some questions if you guys have an interest. Don has a wealth of knowledge, particularly around all things ketones, I believe. But Don, what have you been up to lately? I know you're still out in Florida. I see you're still working out. I saw you doing some HIIT training with the squats the other day. That's a tough workout. I know I've done things like that, and I know it's good work. But
yeah,
continuing to set a good example for what being healthy is about. Have you been? I know you've been doing a lot of biohacking and testing various glucose levels and ketone levels and things like that. Mm-hmm. What is your recent focus been on?
A lot of things. I mean, I really diversify the type of research that today I was outside with the cows all day. So we have a bunch of cows here on the farm. So I was out on the ATV trying to find them. They were back in the back pasture in the it's very warm here in Florida today. It's about 87, 89, but it's supposed to cool off. So this morning I checked on the cows, got some work in, farm work. My parents are visiting, haven't seen them in a couple of years because of COVID and restrictions, stuff like that. Got some ribeyes set out to put on the grill later for them. But in the meantime, yesterday I did a 14-hour work day. So I teach full time. I teach medical students. I teach Ph.D. students. My Ph.D. student gave a really fantastic presentation on the metabolic epigenetic effects of ketones. So we're studying rare metabolic genetic diseases. And then ketones actually function as epigenetic modifiers to influence gene expressions, which have neuroprotective effects. So that's her PhD dissertation. So I do basic science, very mechanistic research. I did my PhD doing patch clamp electrophysiology, where I measure from like brain cells and doing that. And then on the other end of the spectrum, I'm wearing a continuous glucose monitor, and we are running a clinical trial right now in partnership with Levels Health using the Abbott Libre. I'm also using a Dexcom G6. And we have participants who are not diabetic wearing a continuous glucose monitor, doing low carb, keto, intermittent fasting, carnivore, you name it, sort of under this IRB protocol. And we're getting amazing effects on glycemic control. We're looking at non-alcoholic fatty liver disease. We're looking at cytokines, chemokines, hemoglobin A1C, comparing that. We're getting some nice data, and we actually just presented our first cohort of data at several conferences, and we're all going to experimental biology in Philadelphia to present some of that work, but I'm doing the second cohort now. And low carb diets and ketogenic diets are very, very powerful modulators of our metabolic health. So I'm studying that in everything from cells to mitochondria to animal models to human studies.
Interesting. Yeah, it's great you're doing that. And, obviously from a clinical side, I see that all the time. I just see people getting healthier when they cut out, whether it's mostly junk food, I would argue. And I think most of the junk we're eating is, this refined carbohydrate mixed with some oils and things like that. Do you find, so when it comes to the, well, let me ask you real quick, how many cows do you have? How many acres are you on? So I'm just curious about that. It's something I feel more comfortable when I have cows in my proximity. It just makes me feel better. So how many do you have around you right now? I think we froze up. Looks like Dom, can you hear me? I don't know. Does anybody else see Dom frozen up? Looks like his side is frozen.
Hey, I froze, but
yeah, okay.
Hey, Shawn, can you hear me?
Yeah, I can hear you now.
I'm gonna log on to my, okay. I think, is my video frozen?
Your video is frozen.
Okay, well, I'm gonna jump on another device and I'll pop on in 2 seconds, okay? And I'm gonna pop off here, but pop on another device. Okay.
So we got a couple questions in from the folks, so we'll try to get those answered about flipping in and out of ketosis. Becky's asking about that, so I'll make sure we get that question answered. There's a question that Tina has about mitochondrial uncoupling, ketosis for traumatic brain injury. Yeah, we'll look at that too. So where's Dom back in yet? Maybe that's, I don't see him yet. You guys let me know if you see him popping into that on another device. I know he's in, I think, Florida, I think near Tampa, if I'm not mistaken. I know he's got a small little ranch where he's got some cows on there. So if you guys follow him on social media, he's got some interesting posts that I think are helpful. And just a reminder, Brett has, let's see, what do we have in here? Brett saying Tuesday, I think this is a Tuesday mental health meeting. So don't forget that, guys, for you guys that want to do that. Anyway, in case you guys are wondering, I had a ribeye for breakfast. I think I nailed it perfectly. 122 in the sous vide, 1 hour, out of wild, one minute each side, 1500 degrees. Turned out just, I think, perfect. I think just awesome. Oh, your Dom's back. All right, let me see if I can find him. Give me a second, Dom. I'm going to try to ask you to unmute again. Let's see if that's you.
Hey.
So there we go. Got you back. All right, perfect. Yeah, it looks like you're on a laptop or on a phone or a iPad.
Yeah.
Now, so.
Yep. So now I could maybe show the farm a little bit. I don't know if you can see
okay.
But it might be out there somewhere.
Okay, so you're near Tampa, is that right? Is that you're kind of inland from Tampa a little bit? Is that correct, or I can't remember?
Yeah, I'm in Plant City, Florida, which is like between Tampa and Orlando.
Okay, got it. Okay, nice. You got and how long is it? And you work in Tampa? Is that where your office is?
Yeah, at the university is just about a 25-minute drive into. I'm at the College of Medicine there on main campus. It's like the little campus right next to main campus, the College of Medicine. So that's where our lab is. We have the hyperbaric biomedical research lab, which is really a bunch of equipment from the Department of Defense where we create these environmental chambers and we can create like extreme environment conditions. And then we test for performance resilience and sort of neurological resilience inside these chambers. And we run a bunch of
I want to ask, I was flipping through your social media feed, and I know you were talking a little bit about neuroinflammation, and does it have an impact on, because a lot of, I think we're going to be seeing a huge increase in the amount of dementia that's coming with all the metabolic dysfunction we have in society now. I think that's just going to, it's going to be about 10, 20 years down the road. And do we know anything about modifying that, protecting us? I assume ketosis can have a role. I know guys like Dale Bredesen out of, I think he's UCSF, has been showing at least some benefit to ketogenic diets in that regard. What's going on with with brain function, nervous system function around diet, metabolism, ketosis? How does that play a role?
Yeah, good question. Well, systemic inflammation, if we measure, for example, blood levels of different cytokines, so we measure IL-6, IL-1 beta, TNF alpha, all these different, but you know, you really, if we measure high sensitivity C-reactive protein, that seems to be a very good general marker of systemic inflammation. And systemic inflammation will contribute to neuroinflammation. Whenever you have inflammation in your body, it basically will loosen up and alter the permeability of your gut. So the tight junctions in your gut, and also we have the same tight junctions on the blood-brain barrier. So these tight junctions are very sensitive to the level of certain cytokines that are associated with autoimmune diseases and systemic inflammation. So we can kind of make the blanket statement, whenever we have inflammation in our body, in our joints, in our gut, that inflammation will influence the permeability of the blood-brain barrier, and that will contribute to neuroinflammation. So this can be caused by a variety of different things. Obviously, our food system is contributing to that, and that's where I think the utility of the carnivore diet comes in to improve that. And different viruses can cause neuroinflammation. You know, shingles virus, herpes simplex, different Lyme's disease can cause that. And nutrition is a really powerful lever that we can pull to mitigate some of that.
Yeah, I consistently see low markers of inflammation on people on this diet, particularly, you know, it's interesting. In the short term, you know, if you check people a week or two in, sometimes they'll see a bump in their C-reactive protein, but it seems like after a couple of months that levels out and then they end up with very low numbers, you know, and so I'm not sure what the initial, maybe it's just the initial stress on the body from switching to a new dietary system, and then we actually have kind of an equilibrium reach. We seem to be at a low inflammatory state. And it's interesting you mentioned, you know, I've heard this before, you know, leaky gut equals leaky brain. I mean, it seems like if one membrane, leaky mitochondria, leaky cellular, you know, permeability all across the board. And so it seems to all be correlated. What are you finding with, you know, again, I know I've touched on this before, but ketones themselves, the beta hydroxybutyrate or the other ketone bodies, or is it just the, you know, the lifestyle things that you do to get into ketosis that are making a difference? You know, and I guess there's a question for you that somebody asked. Is there a problem being chronically in ketosis? I know we see that being touted as a bad thing. You know, you can't be in ketosis all the time because you're going to have problems with, say, cortisol rising and things like that. What are your thoughts regarding cycling in and out of ketosis? Is that beneficial? Is it necessary? What are your thoughts?
Very good question. Yeah, you know what? I should probably address this question more often than I do, and not sure I actually have. But in the epilepsy community, I just gave a talk to the NIH and Johns Hopkins, and they are under the, you know, in their world, the ketogenic diet is unnatural, and it's not something that you would want to do chronically unless you have a condition that is managed by it. And that would include different seizure disorders, metabolic disorders like glucose transporter type 1 deficiency, you know, pyruvate dehydrogenase, a bunch of inborn errors of metabolism that go by long names, or epilepsy. And I am somewhat of the opinion that staying in ketosis 100% of the time was probably not natural. And even a ketogenic diet, which is, you know, 90% to 80% fat, is probably not natural either. I would say that, except for a few remote populations. So, you know, my ketogenic diet is much higher in protein, upwards of 50% on some days, you know, 30 to 50%. And I will occasionally, you know, go out. I'm always measuring or registering, I should say, some level of ketosis throughout the week. And sometimes I'm higher, sometimes I'm lower. But I like to achieve it not necessarily with a strict counting out my macronutrients and the ratios and things like that, but simply just doing like intermittent fasting throughout the day. Like today, for example, and then I'll just start eating around 4 o'clock. I'll have some steak, maybe some fish and some other things. But I'm of the opinion that chronic ketosis is not something that should be done all the time in people that are healthy, unless it can be very effective for people that are insulin resistant, that have weight to lose. But once you've gotten a hold of and managed your condition, whatever you're using the ketogenic diet for, you can back off of it and you don't need to be in a state of ketosis. I mean, there are many benefits to having elevated blood ketones, because ketones are a hormone-like signaling molecule that have their own effects, even on suppressing inflammation and even epigenetic effects. But this is a very nascent area of science, and we are sort of at the edge of that, studying it in animal models to even figure out if this is relevant to humans. I mean, we definitely see some interesting effects on physiology, on gene expression, on neuroprotection, when we put animals inside environmental chambers, and push them with a protocol that would typically kill them. If they're in therapeutic ketosis, they can survive extreme environments that would otherwise kill the animal. So these are observations that we made 10 years ago, and it got me super excited in this field. So we are developing different ways to achieve ketosis and induce it and sustain it under a variety of different paradigms. Some people cannot, some people are unwilling, I should say, unwilling or unable to achieve and maintain ketosis. So we've developed various technologies, I guess you would say, to achieve that, mostly for the military operations, but we're also, using some of these things outside of military.
Yeah, your diet is very similar to mine, in that the protein content. I've always felt that protein was a very, very important macronutrient, and I like I said, like you, if I'm eating ribeyes, I'm probably 30% protein. If I'm eating sirloins, I'm getting closer to 40-50%, and so I do that pretty frequently. There's a question about some people just have a hard time seeing ketones, and I know that there's this sort of balance between production, utilization, and waste. And I know I've seen some people that have been chronically on ketogenic diets, and yet they show minimal ketones in their blood. Is it just an adaptation to becoming more efficient at utilizing them, that they don't show up as a waste product? Or what's going on when people can't? Because I rarely see high ketones. I mean, I think the highest I've ever seen is like 1.5 millimolar, and that was, I don't do a lot of fasting. I mean, usually when I check, and I don't check it that often, but usually it's 0.3, 0.5, something like that. Nothing very high for me. Why is that for some people? Is it just we've adapted to it?
Yeah, that's a good question. The ketones that you're measuring in your blood are a product of ketone production and ketone utilization. So especially in athletes, what we see is that there's a very high level of tissue uptake of ketones. So once you are chronically adapted to ketosis, you actually upregulate the transporters, which are the monocarboxylic acid transporters and also the ketolytic enzymes. So these are things that we sort of measure in the lab. Like in animal models, you put like a rat or a mouse on a ketogenic diet, and you'll see a 50% upregulation in the proteins that are associated with MCT transporters. So there's more ketones being dumped from the blood into the tissues. So that could decrease the level of ketones that you see in your blood. I also use a Biosense breath ketone meter right here, and when I'm fasting or I'm in a deep state of ketosis, especially if I'm in like a calorie deficit, what I experience is that I have a very high level of ketone utilization. And I know this because, I can be at my desk not moving, and my ketones really rarely get above 2, but they could be, for 2, that's high for me. And I could go for a walk and do some stuff around the farm, and they'll be back down to like 0.3. And I am not decreasing ketone production, but I'm actually increasing ketone production because I'm probably burning more fat. Ketone production will be proportional to the amount of fat that you're burning, but I'm also disposing of ketones, similar to, disposing of glucose after a meal if you go for a walk. So that there's some things going on there. And also, it's very individual. Some people, experience high ketones and some people don't. Someone who's very sedentary and insulin resistant that goes on a ketogenic diet, I've seen ketone levels climb in the 5 to 6 range, but I personally feel best at about 1 millimolar of ketones in the blood.
I guess it's analogous to, glucose disposal. I mean, if your glucose is high and you exercise, it's going to go down. And I know, like, for instance, this morning, I happened to check my glucose. It was 75 when I woke up. I did a 20 minutes on the exercise bike as part of my morning wake up. And then I checked it again. It was 49 and my glucose was, I mean, if a doctor might see me and give me some stat orange juice or stick a dextrose IV in me or something like that. But let's talk a little bit about that low blood glucose in the context of a ketogenic diet. I mean, I know, back to whenever Cahill was doing his research, you often register blood glucoses in the 50s and stuff like that. Are you concerned about that for someone in this situation? I know it can be troubling for, say, a diabetic and they keep having these hypoglycemic episodes, but I'm sitting there at 49 and I'm fine. I don't feel bad at all. I don't have any symptoms of hypoglycemia. So should I be concerned about that?
No, absolutely not. Especially if you have low, normal levels of insulin. Some people will secrete a proportionally higher amount of insulin in response to certain amino acids, and that can drop your blood glucose. But if you're asymptomatic for hypoglycemia, which you typically are if you're on a keto diet, carnivore diet, or even just a low carb diet, what happens over time is that the relative changes in your glucose could, if you have a very large drop in glucose, then that could be perceived as hypoglycemia and you can get shaky. It activates your sympathetic nervous system. But what we see, for example, in like type 2 diabetics who could be, their normal glucose could be 180. And if they drop down to 120, they could experience hypoglycemic effects from just a drop to 120, even though it's above normal, because it's a relative change. But over time, and I've seen this in type 1 diabetics who follow a ketogenic diet, they could drop in the 40s and be asymptomatic. So if you have a type 2 diabetic, and over time they drop their blood glucose, then their physiology adapts to that level of glucose where they have a higher, glucose utilization, better transport of glucose, better glucose efficiency in the brain over time when their glucose goes low, and they keep it low for a period of time. And then, once they do that, they basically have metabolic resilience. And if their glucose levels drop, especially if their ketones are elevated, they can be hypoglycemic under clinically, they can be hypoglycemic clinically, but asymptomatic for hypoglycemia. And we see that in people that are adapted to ketones. They're keto adapted.
One thing you mentioned was, utilizing, going to exercise and seeing your ketones being utilized. I remember seeing a talk from a guy named Nadir Ali, who's a cardiologist, and he was making the assertion that it's more the brain utilizing ketones than the muscle, and that the brain is the largest consumer of ketones. Is there some truth to that?
Yeah. So, over time, once you get on a ketogenic diet or go into fasting ketosis, there is a high uptake and utilization of ketones in the skeletal muscle. But over time, the skeletal muscle has a very robust adaptation to using fatty acids for fuel. So skeletal muscle will dramatically upregulate, mitochondrial biogenesis and fat oxidation enzymes and things like that. Whereas the brain's capacity to do that is like almost non-existent, to use fatty acids for fuel. It can use like short chain and medium chain fatty acids, but pretty much long chain fatty acids won't cross the blood-brain barrier. So that is correct. In the beginning, you'll have a transition from high glucose or high ketone utilization in the skeletal muscle to lower ketone utilization, but higher fatty acid oxidation in the skeletal muscle. And you could have about a 200 to 300% increase in fat oxidation in the skeletal muscle, once you've really induce and sustain a very low carbohydrate diet over a period of time. And then it'll downregulate the ketone utilization to essentially spare the ketones for the central nervous system.
When you say over a period of time, what kind of time frame are we looking at? Because we hear a lot of athletic studies, they'll do a 3-week trial on low carb versus high carb, and they'll invariably show greater power output in the higher carb group. And then, a lot of people say, well, 3 weeks isn't enough. What do we have a handle on what's enough, particularly when it comes to athletic performance for, the appropriate adaptations to occur?
Yeah, another good question. I would say like months to years. I would give it a good 3 months. We see changes continuing to happen after 3 months. And since we still see, upregulation and inducible changes after 3 months, I would say it would at least be beyond 3 months. And personally, when I did a ketogenic diet, I started with a clinical ketogenic diet, and there's a learning curve to it, but the more I did it, the more it was working for me and the better I felt. And then my strength kind of returned over maybe 3 to 4 months. It took, to really get my strength back to normal, I think. But once, you've adapted, I do feel that there's like a metabolic adaptation that takes place similar to muscle memory. Once you've trained your metabolism to be a very high fat oxidizer and lower glucose utilization, and you go off of the diet and then you return, I think your metabolic flexibility is greater. So we actually published a study in rats. We put them on ketogenic diets for a period of time, and then we take them off, and then we put them back on again. And the rate at which they achieve ketosis again is about 50% faster. So it's basically showing, and the level of ketosis will be proportional to fat oxidation. So what that was basically telling us is that when you switch into a standard diet back to a ketogenic diet, after they had been on a sustained ketogenic diet for a period of time, their fat oxidation machinery is much higher, and they're able to make, to burn fat and to make ketones. So ketones are a byproduct of beta oxidation of fatty acids in the liver. And if you can achieve that, a 50% higher increase in ketones, the latency to a certain time point, if that is reduced, that's basically showing that you have sustained metabolic flexibility, after you've done a ketogenic. No, in rats and mice, all this happens much faster in humans. So a mouse metabolism is about 6 to 7 times faster, and a rat is like 4 to 5 times faster. So I kind of extrapolate it from that point of view. You could fast a mouse for like 24 hours, and it can lose like 20% of its body weight. Obviously this doesn't happen in humans. So things are faster. I mean, that makes it convenient to do these metabolic studies. But the translatability as far as the temporal or the time scale is something that we don't fully know, because what we do in mice and rats would not be possible to do in humans. We can't take their brains out and look. We can't, look at the blood-brain barrier and do elaborate, liver analysis and muscle analysis that we do in mice and rats.
Yeah, that's one of the limitations to human studies. We can't kill all our subjects at the end and cut them open. You started to talk about, you have animals that are people or whatever that are resistant to going to ketosis for whatever reasons, and you said there's ways to sort of tip the lever that way. Are you talking about exogenous ketones, or are there other things you're doing to sort of facilitate ketosis for those people that don't want to or have a hard time doing so?
Yeah, we call that, I guess it was Mark Madsen at the NIH that created this term metabolic switching. And we actually included that term in the title of one of our publications, but it was in an aged rat model. So in rodents that are aged, their metabolic flexibility is somewhat decreased. They don't go into ketosis as fast. But if you do intermittent fasting, you can trigger that metabolic switch per se. So if someone's doing a ketogenic diet and they simultaneously do intermittent fasting or time restricted feeding, those two things are pretty synergistic. And as we age, that may be a way to further augment our fat oxidation. So going a period of time, eating within a 12-hour window, you don't have to fast for like 18, 20 hours. So what I like to do, and I don't even do it every day because I'll just lose too much weight, but I'll eat within a 12-hour window, or sometimes an 8-hour window, depending on my work schedule or whatever. And that's when I'll achieve like my highest level of like fat oxidation and ketone production. And I think that the breath ketone meters, I've measured breath thousands of times, and I've measured blood ketones like thousands of times. The breath ketones are a better indicator of fat oxidation, I believe. And they're measured in, the Biosense uses the ACEs, and I can really pin that when I'm fasting or just eating like one ribeye a day. Like sometimes I'll do that. And that'll be when I achieve my highest ketone levels, when I combine carnivore ketogenic diets with intermittent fasting. And that'll be the metabolic switch that can help people, if they have a hard time getting into ketosis, achieve a higher state of ketosis.
You mentioned a preference for 30 to 50% protein, which again is fairly similar to what I do. Some people will say that, because you're eating that much protein, you're gonna tend to lean more towards gluconeogenic pathways, and that inherently is bad for some reason. I tell people then don't go to sleep or something if you're worried about gluconeogenesis. But I mean, and honestly, I can still do very good with the glycolytic stuff. I mean, I still train very hard in a glycolytic fashion. I'll see my blood glucose going quite high, particularly after intense exercise for at least a short period of time. And so, I think, my liver's obviously breaking down glycogen and spewing that out while I'm exercising. But are there any concerns about protein? And is gluconeogenesis somehow a bad thing for us that we should be worried about? Because there's a lot of people that say, by going low carb, you're driving gluconeogenesis, and that's somehow stressful to the body. What's the deal on that?
No, gluconeogenesis is natural and probably optimal for an athlete, because if you upregulate the gluconeogenic pathway, gluconeogenesis will be, if you're an athlete, you activate that system preferentially if you are doing higher glycolytic activity. So some people will say, like, if I do a ketogenic diet, how do I train my body to be, to function in a highly glycolytic state where I'm putting out a lot of energy and doing sprints and things like that? If you train in the context of, being in a state of ketosis, and you do that training, you may see your performance decrease initially, like you most likely will. But if you train under those conditions, then you will forcibly adapt your body to liberating more glucose for fuel when you need it. So through the metabolic adaptations, you can upregulate glycolysis in the context of doing high intensity exercise over time. And that's good. That's going to take months to really train your metabolism to do that. So that elevation in glucose will be advantageous in regards to, you could be, your fat oxidation's high, your ketone production could be high. And if you train under that condition in a low carb state, you can train to upregulate glycogenolysis and gluconeogenesis to ensure you have multiple fuels going to your muscle in the context. And if you have done the majority of your training like fasted or low carb, and you're a competitive athlete, you could then titrate in and add small amounts of honey or carbohydrates in and around your training if you're worried about that, or you can experiment with that. I don't need to do that because I'm not really like a professional athlete or anything like that. But when occasionally I will do that and just add a little bit of small, I'm talking about like 15, 20 grams of carbs, not like 100 grams of carbs, something like that. A small amount can go a long way in that context.
Yeah, and that's very similar to my experience, quite honestly. It took me a couple months to kind of adapt, but I kept pushing through, I kept grinding through, and I was able to, at least on the concept to set world records in this very low carb, carnivore state. I guess the counter to that be, well, why jump through all these metabolic hoops to produce glucose feed and gluconeogenesis when I can just swig down some sugar water and get me there the same thing? Is there an advantage to that? You said it's optimal. Is there a potentially deleterious effect to consuming, relying on carbs ingested? Are there inflammatory implications? Are there reactive oxygen species implications? What are your thoughts around that?
Well, it'll be completely dependent upon, the amount of calories you're consuming of carbs. So I think it's always important from my context to keep your body hungry for sugar. So you don't want spillover of sugar where you're ingesting carbohydrates, you're dumping insulin, and then you have a postprandial or post-glucose ingestion decrease in your glucose from insulin, and then you'll shut off ketone production too. So you'll be hypoketotic and hypoglycemic, which will give you a headache and will tank your performance. I notice this actually if I am in a low carb diet or a fasted state and I consume a large dose of ketone esters. So that's why I'm not a big advocate of jacking up ketones real high, just because I'm also not an advocate of jacking up glucose very high. I think the sweet spot for me is just getting ketones mildly elevated, 0.5 to 1.5 in that range. For the average person, if you're using this to manage a metabolic condition or epilepsy or cancer or something like that, maybe 2 to 3 or 4 would be better, the ketone levels. But yeah, I think when it comes to ingesting carbohydrates, the dose makes the poison. And I think if you're really slamming lots of glucose, then you become so glucose dependent, then you need that for fuel to function. And I noticed something that's very interesting is that if I switch to eating carbohydrates for a period of time and I don't eat them and I exercise or I go hiking or biking or something like that, I get flat really quick. I sort of train my body to use glucose for fuel. Even in like a week or so, this could happen. We were on vacation and I was eating more carbs than normal. And I went a period of time without eating. And it's almost like my body deflated because I then kicked on the glycolytic machinery, and then I basically catabolized all the skeletal muscle glycogen and liver glycogen, and then I felt hypoglycemic. I felt super flat. Whereas if you are fat adapted, it has a glycogen sparing effect, and you spare glycogen because you are pulling off your fat stores for energy much more efficiently and making ketones, if that makes sense.
Yeah, it does. We see that in context of say an oral glucose tolerance test. If you're chronically on a very low carb ketogenic or carnivore diet, you're going to probably fail an OGD, oral glucose tolerance test unless you spend 3 days a week or whatever consuming a amount of carbohydrates. And I'm sure what that is, 100, 150 grams a day for a while. So you kind of switch that metabolism back to a more glucose, dependent situation. Is there any difference, just about the different types of saccharides and different types of sugars, glucose versus fructose? I know there's a lot of controversies, fructose driving non-alcoholic fatty liver disease. Any concerns about those two different variations of different ways to get sugar?
Yeah, I think, again, the dose will make the poison. I would never, I'm not a fan of drinking any kind of carbohydrate beverage, right? I think fruit is fine in moderation. I have berries almost every night. On many days, I'll just have, steak and eggs and fish. That'll be the only thing I eat all day. And then I'll have the only plant-based thing I'll have is maybe some blueberries, at nighttime or something like that. And if you're eating that in the context of a high fat, high protein meal, that has very, very minimal effect on glucose levels. Whereas, if you eat it in a fasted state, I'll see my glucose pop up and then maybe come down a little bit lower than normal. And that could maybe elicit an insulin response or something like that. But that's pretty minor. But yeah, I don't know. Does that answer your question?
Yeah, sure it does. I had something, it just went out of my mind. When you have, well, let's see where we're going to go with that. I forget. Sorry, Don.
So I tend to, if I do
yeah.
have carbs, I do it like in the evening, and it's sort of like just a snack, and I make like a little mousse pudding. Sometimes I have some keto ice cream or something like that. So I keep a little bit in my diet, and that's just as like, you know, a reward mechanism, and it's something that I enjoy eating like at nighttime. And oftentimes on the days I do intermittent fasting, I tend to get my calories in later in the day. I don't see any negative consequences to this in my sleep or performance or how I feel or anything like that. So.
Yeah, that's what I was going to ask you about is the circadian biology. There's a lot of, thought about, when we're supposed to eat, timing a day. I personally do better with earlier meals. I had breakfast at 7 a.m. I'll probably have another meal at 2 p.m. and that'll be it for me. I find it results in just better overall, whatever for me, sleep and things like that. But what do we, and the other question, so what do we make about circadian timing of meals? And the other thing is you mentioned, compressing your eating window. Is it just a caloric restriction? You listen to Lane Norton, he says the only reason that works is because of calorie restriction. Is there a benefit outside of calorie restriction? If we have isocaloric consumption of calories over a longer window versus shorter window, is there an actual benefit there?
Yeah, so I'm of the opinion that 2/3, it's hard to put a number to it, but I don't think all the benefit is not a result of calorie restriction. I think that's clear. I think they're, time restricted feeding protocols that show that, eating according to circadian biology has beneficial effects. But I do think that for weight loss and weight maintenance, the primary benefit of time restricted feeding is that it does allow people to more easily produce a calorie deficit. But I think there's big advantages to eating, in a compressed window when it comes to sleep and things like. But then again, if you have a fast metabolism, eating within a compressed time frame may not give you enough calories, may not give you enough protein. For me, I do like to spread my protein out. Like yesterday, I just had 2 meals. I was so busy. I ate in the morning. I worked like a 14-hour day, and I literally didn't eat anything 14 hours after. I had to pick up, I had to go to the airport at like 10 o'clock at nighttime, and I was eating kind of on the way to the airport. Had like some sardines and a hamburger patty or something like that. But I felt super great. I felt like supercharged all day. And I tend to gravitate, because of my work schedule, to going back to eating breakfast, which is usually just like some scrambled eggs and some beef. And then I'll basically, I'll be so involved with my work for 12 hours, I don't get hungry. I'll make some coffee and I'll put it in a mug and I'll sip on that throughout the day. And I try not to get much caffeine after about 1 or 2 p.m. in the afternoon. And then I'll just be cranking out a lot of work. And then after work, sometimes I like to hit the gym, usually around like 5 or 6 o'clock. And then I'll eat my second meal. And then I'll have maybe a little bit of snack in the evening or something like that. That has been the protocol that I always go back to. Unless I'm like teaching and I have to teach real early, or I got a lot of stuff front loaded in the morning. And then I feel best. I feel mentally, I have the best mental clarity and cognitive function in a fasted state, and I'll do that in the morning. I'll fast throughout the morning a couple times a week, under certain conditions. Depends on my teaching schedule, but I feel fine doing that too. But I like to mix it up a little bit, but it's really highly dependent upon my work schedule, which can be pretty variable, especially with traveling and things like that.
Yeah, traveling always makes it very tough. There's a question here about traumatic brain injury, and I don't know if you delve into that much or have had models on that. They're asking about how do we deal with that from a nutritional standpoint? Are there supplements and things like that, perhaps exogenous ketones, ketogenic diets? Do you have any insight into traumatic brain injury and how to perhaps best deal with that based on what you've seen?
Mm-hmm. Yeah, we wrote a review with some of our academic colleagues that basically is a systemic review and meta-analysis on all the animal models and whatever human data was available on this topic. And I've communicated with quite a few people that have gotten brain injuries, concussions. You know, a couple people come to mind. Peter Attia's brother, actually, Paul Attia, he's a very good friend of mine. He had a pretty bad bike accident, you know, kind of help him recover from that. Yeah, you know, traumatic brain injuries, as some guys, you know, in the military have gotten that and communicated with them. If you have a penetrating traumatic brain injury, about 80% of those guys have seizures. So in that context, the ketogenic diet is not only useful, it's almost a standard of care. It can be, because some of those seizures are also drug resistant. So, you know, following something like a modified ketogenic diet that's supplemented with things like MCT or exogenous ketones to further elevate your ketone levels. Of course, we know a number of different brain nutrients like DHA and EPA, or, you know, omega-3 fatty acids can be very helpful for restoring and maintaining, you know, brain function, but also repairing the blood-brain barrier. I'm also a big fan of things like taurine and under certain conditions, GABA, or the drug phenibut can be useful for some people because it's basically, you know, phenylated GABA can get into the brain a little bit easier. Acetyl L-carnitine, alpha lipoic acid. I think these things they all provide some benefit in animal models, and I think when they're used together, I think they can be very helpful. Magnesium, too. Magnesium. So when you have traumatic brain injury, the neurons dump a lot of a neurotransmitter called glutamate. And if you have lots of glutamate being dumped from cells, that can cause excitotoxicity and can cause the cells to depolarize, and it contributes to excess calcium, and that can actually trigger a seizure. But high levels of magnesium, magnesium threonate, BioOptimizers makes magnesium breakthrough product, which is like a half dozen different forms of magnesium altogether. I've taken that and then gotten blood work, and then my blood levels of magnesium are very high, so that's very effective. But there's also things like the product Calm, which is like magnesium citrate, magnesium malate. So yeah, I think ketogenic diet, exogenous ketones, omega-3 fatty acids with a good dose of EPA and DHA, alpha GPC could be beneficial, magnesium, all these things I think can be very helpful for brain injury patients.
You mentioned glutamate, so that always makes me think of Dr. Thomas Seyfried, who I know you've communicated and worked with in the past. Any updates on cancer with regard to diet? I know he's been still plugging away with that. I don't know if you still have a relationship with him, or is there any more data we know about cancer and ketogenic diets or diets in general?
Yeah, so there's a glutamate-glutamine cycle, right? So the astrocytes will suck up glutamate and actually convert glutamate to glutamine, which is a neutral amino acid that gets pumped back into the neurons, and then it's converted back into glutamate. So it's this cycle. Is one of the classes that I teach in neuropharmacology is the glutamine-glutamate cycle. And for cancer, I know when we get brain cancer or we have radiation of a brain tumor, something like that, that dumps a lot of glutamate and glutamine into the extracellular space, and that could be more fuel for cancer cell growth. Tom Seyfried has done some studies using the glutamine blocker DON, which is an analog of the amino acid leucine, actually. And it blocks glutamine metabolism. So the only thing is that that compound can be toxic. So the only way to really target, when we go on a ketogenic diet, blood glucose drops, insulin levels drop, and the efficacy of a ketogenic diet for cancer would largely be a product of reduced glucose availability, but also by suppressing insulin, that suppresses insulin-associated signaling, like, you know, AMP kinase goes up, mTOR goes down, IGF-1, things like that, and various growth factors are affected. So that's, and then when cells are starved of glucose, many different cancer cells can drive fermentation as a means to make energy through glutaminolysis, which is the metabolism of glutamine essentially for energy, for ATP. And we don't yet know effective ways to block that. So we are interested in targeting that. We've used a couple different compounds to try to do that. I mean, some of the more, non-toxic ones would be like sodium phenylbutyrate. It's sold as buphenyl. It's used for urea cycle disorders. So that actually can bind up glutamine, and then you pee it out. So that could probably lower glutamine, probably maybe about 10 to 20%, and that could be beneficial. But there's experimental drugs that are being used now in basic science research, but these things are in my opinion, there's too many side effects associated with them. Whereas something like sodium phenylbutyrate, which is sold as buphenyl, typically at about 5 to up to 20 grams a day, I think could be helpful. We've used it in some different model systems, and I think it could be part of a comprehensive metabolic strategy to target cancer. We are not, in the context of advanced metastatic cancer or glioblastoma, we are of the opinion that, the standard of care probably helps very little. Although in the context of, ketogenic diet or metabolic approach, that may augment the effects of standard of care to make it work better. I know Thomas Seyfried is adamantly opposed to the standard of care, and that's where I tend to differ with him. I'm a bit on the fence, and it depends on the individual and the type of tumor that they have, and whether that's, there's genetic variations in different tumor types. So I think you have to approach each situation from the context of that individual, and not say, completely eliminate, don't do standard of care. Under some situations, I think the standard of care can work and be life-saving, but I think it'll always work better in the context of going into it in a fasted state, going into it following a ketogenic diet, and using other metabolic-based drugs like Luke Cantley has the PI3 kinase inhibitors, metformin can offer some synergy with the ketogenic diet too. These are things that we study in our lab, in animal models at least.
Yeah, I've seen, anecdotally, a number of people that have gone through standard of care, cancer therapy, chemotherapy, and on carnivore ketogenic diets, and they often report that the side effects have been minimal. They've tolerated it much better, and even the physicians are remarking how well they're doing relative to other people, which I think is interesting. I wanted to go back to, you mentioned early on, you guys now have a new study in process looking at a number of things, and one of the things you talked about was gut permeability, and so we have the tight junctions. I'm just wondering how you're assessing that. Are you looking at zonulin levels, or are you looking at some other way to, or are you even tracking gut permeability in a sort of a scientific way, or is it, I look at the work of guys like Alessio Fasano I think he's out of Boston Children's, looking at the relationship between gut permeability and autoimmune disorders, and there seems to be a pretty clear link there. Are you measuring the gut permeability, and if you are, how so?
Yeah, that's something that we really want to do. We have a big microbiome center here, but to be honest, the majority of people, and we'll have about 50 or 60 people in our clinical trial right now, they do not have problems, with gut permeability going into, or actually it improves so much with low carb and ketogenic diets, it becomes sort of like a non-issue. We have done studies, in extreme environments where we have in our -80, we have lots of fecal samples before the exposure and during the exposure and then after the exposure. So we have not analyzed that. As we wait, the tools that become available to look at microbiome changes become increasingly less expensive as the technology advances. If we would have analyzed these samples, 5 years ago, it would have been like, a big 6-figure budget, whereas we can do that for probably a third of the price now, just because the technology is getting much better. I have communicated with quite a few groups, even international, outside the United States. Paleomedicina, for example, they use the PEG 400 test, I believe, where you drink that and then you look at urinary levels of that. So we actually went there to Budapest, Hungary, to their clinic, and they pulled all the files, of their patients, and they showed me patient after patient that they put on a paleo keto diet. If you just check them out, Paleo Medicina, and look them up, they showed me patients with type 1 diabetes, type 2 diabetes, brain tumor patients with all sorts of injuries, and they showed me their gut permeability results from these tests over time. And just going to a pure meat diet, just eliminating, and I think, they are very strict in their protocol of what they allow, in their diet. I don't know, are you familiar with them? Or they would be a good group to interview.
Yeah, I've interviewed Sophia and Chaba several times. I'm very familiar with them.
Okay,
And one of the concerns, they tend to be fairly calorically restricted. And I think, outside of cancer, like for me, I couldn't live on the amount of calories they would recommend. It's just not enough for me. And I've kind of asked her about that. But yeah, they've got some very compelling and interesting data. But like I said, they like you said, they use that PEG 400, that polyethylene glycol study to assess that. And it seems to be pretty compelling. It's pretty interesting for sure.
Yep. Yeah, I mean, you can't argue with their data. I became really convinced when they pulled files and started showing me blood work before and after. I do think a lot of their success probably comes from the calorie restriction component. And I know my colleague, Tom Seifert, is very adamant that, in the context of cancer, calories need to be restricted. We did a study in our mouse model, and we did not want to restrict calories. So we just did a ketogenic diet and found that weight was pretty stable, and we got a remarkable anti-cancer effect. We published that, or when we tried to publish it, then the reviewers they were saying, this is a calorie restriction effect. So we took the standard diet and then restrict calories, I think 20 or 30%. And as you can see, the body weight comes down. And that also decreased the metastatic spread of the cancer cells, but it did not work as well as the unrestricted ketogenic diet. So just to restate that, a calorie restricted standard diet with carbohydrates, and it wasn't even high sugar, it was like, fiber and everything like that, was not as nearly as effective as a ketogenic diet that I designed. It was sort of a custom ketogenic diet that was an ad libitum diet where they could eat as much as they want. And it had higher levels of omega-3 fats. It also had a pretty high percentage of medium chain triglyceride as a fat source in that diet.
Yeah, that's pretty powerful. I mean, clearly, and there are some big proponents of, calorie restriction, protein restriction, as well as a means to, i.e., live longer or not get cancer. Do you have any concerns around that? If you listen to guys like David Sinclair out of Harvard, he's saying, minimize protein, certainly minimize animal protein due to this effect on mTOR and other factors. And it's mostly based on, I think, some rat studies. There's not really good human data on that. Do you have any concerns about eating a higher protein diet in the context of cancer or aging?
I don't. I'm of the opinion that the more muscle you have, that muscle can not only be the fountain of youth in the context of function and how we feel and how we look and things like that, but muscle also produces myokines, and muscle produces hormones and things that can affect brain function. We know this now. So the more muscle we have, that is metabolic currency, and that is extreme. Probably one of the most, the amount of muscle we have and the amount of strength we have will directly correlate with longevity. And that would require, in my estimation, a protein level that's at least 1.5 grams per kilogram, upwards to 2 grams per kilogram, is sort of somewhere in between that. Going above 2 grams per kilogram, which I do quite often sometimes, may not offer any additional benefits, and I'll say that. But I think they're advocating for something like 0.75 grams per kilogram, which is like half of my minimal threshold for protein. So I'm an advocate, and I feel that protein is absolutely necessary to build and maintain muscle, which will directly correlate with longevity.
Yeah, I'm definitely like-minded on that. You know, if nothing else, better function. And we've seen, I think, I don't know if you're familiar with work of a guy named David Barr. I think he's out of
Right.
UC Davis, and he's done work around longevity, I think, and looking at rats. And I think they've given them rapamycin. And you see, you know, if you give a blanket rapamycin, they just become sarcopenic because it shuts down muscle protein synthesis. And so you might live longer, but you're in a sarcopenic, frail state. Whereas if you exercise and eat enough protein, you live longer, but you have much better function. I think that's the way I look at that. I don't know if you have seen something similar.
Yeah. And, you know, exercising basically increases the utilization of protein for muscle building and sustaining muscle. So, you know, and I think the power, like exercise is a huge lever, right? And exercise will basically augment protein metabolism in a way to enhance skeletal muscle protein synthesis. And if you're giving your body suboptimal levels of protein, then you're not maximizing that. But if you're dumping huge amounts of protein, and then you have like protein spillover, amino acid spillover, that could potentially, but I think that threshold is high, and it's probably well above that 2 grams per kilogram. I mean, at one point I was getting, you know, 4 grams per kilogram of protein back when I was younger. I mean, I was literally getting over 500 grams of protein a day in and day out. And it didn't seem to have any negative consequences. My liver enzymes were on the high end of normal. And then when I went to ketogenic diet, especially when I calorie restricted a little bit and lost a little bit of weight, then my liver enzymes were on the low end of normal, if that's any indication. But I think that's normal liver metabolism if you're consuming a lot of protein. Your body's just doing what it's meant to do.
Yeah, I think the same thing we see with kidney function. We'll see higher BUN and higher creatinine, you have more muscle mass, you're eating more protein, and that often confuses physicians even because they just assume your kidneys are shutting down. It's like, no, it's just normal high protein metabolism. One last thing I know, you know, with the mice studies, I know I talked to Don Layman before. Well, I mean, obviously you talk about the high protein mice. Jose Antonio, who's kind of one of your neighbors, I think he's in that area with you. He's done the high protein feeding with the athletes and shown that guys eating 500 grams a day, no deleterious effect to kidney, liver, any of that stuff, even over a year or two. But Don Layman pointed out to me that, you know, the most lab rats tend to overeat. I mean, you know, they just tend to overeat by 30%. And so when you calorie restrict those guys, you're just putting them back to a normal level of calories. You're just taking them from oversupply to normal. And that's probably where the majority of the longevity effect is. It's not that they're, you know, truly calorie restricting beyond their normal needs. Is that a fair statement, you think?
Yeah, and it depends on the strain. Like we use Sprague Dawley rats, Fisher rats, C57 black 6 mice, VMDK mice. Pretty much all of them will overeat. The VMDK mice are like your perfect couch potato mouse. So we got them actually from Thomas Seyfried. And they will eat and just become like fat little balls over time. And they are just metabolic syndrome off the charts. And if you calorie restrict them or put them on a ketogenic diet, they become like super mice. So you're basically, and I think it's a good analogy to like the average, you know, person in the US, you know, who's just, they're inactive or hypoactive and they're hypercaloric. And that's creating a scenario where you're just causing insulin resistance and metabolic syndrome. And a little bit of calorie restriction can go, you know, a long way. Yeah, with mice, you know, 25% calorie restriction does amazing things actually to their libido. So when we have many different breeding protocols where we have to breed up different strains of mice, and when they stop breeding, we'll do like intermittent fasting or just restrict their calories by like 25%. And then their breeding is sparked, their libido goes off the charts. So it's in mice, if they overfeed and become fat and their metabolic profiles are tanked, then they become like completely, it just like crushes their libido and their ability to breed and maintain the colony. So that's an interesting observation we've seen many times in many different strains.
Well, Dom, you're a wealth of knowledge, and thank you for spending some time sharing with us. I just want one last thing. I mean, just in a nutshell, I mean, if you were to say like general sort of heuristics on sort of life and longevity, restrict carbs to some degree, eat adequate protein, maybe do some resistance training. Is there anything more you'd add to that? Is that a pretty decent sort of summary?
Yeah, I think, get moving every day, obviously, but I think it really is important to lift really heavy and hard, just twice a week. I found, the same amount of effort that it takes to build muscle is quite different than it takes to maintain muscle. So maintaining muscle is pretty easy, but I think that's one of the biggest levers we can pull. And you don't have to be on a ketogenic diet, but just dropping carbs, below. If you're on a standard American diet, just start with like 100 grams a day. That's actually what we do in the clinical trial. And then we move to like 50 grams after 3 or 4 weeks. And that transition seems to be very, very good in regards to compliance, because when we rapidly try to put someone on a very carb restricted diet, then there's sometimes they get side effects, and sometimes we just don't get good compliance. So we've learned over time, and my medical MD colleagues, that transitioning someone has worked quite a lot better than abruptly putting them on a very low carb diet. So that could be important. I would have not seen that too. But she convinced me over time by just showing me the data. It's like, here's what happens when we, transition, and it's like you get a 50% better compliance.
Yeah, I can see that for sure. I guess the only exception is some people that have these real addiction issues and they have to abstain rather than moderate. And you have some of those people in the mix there. Dom, tell us where to go. And I'm interested to see how this clinical trial, so hopefully we'll get that. You'll be able to share with us the world when you get the results of all these neat things you're doing. Where do people go to find out more, Dom? Where's your social media? Do you have a website, all that good stuff? Maybe, I don't know, have you written a book yet? I don't know. It seems like you should if you haven't. So I don't know.
Yeah, I get, it's different. It's difficult. I'm full-time teaching and I supervise many research projects at the university on top of all the committee work and stuff. So the book is something that I'm entertaining, but I do have an information website, ketonutrition.org. So ketonutrition, all one word, .org. And we have a blog on there. So every 2 weeks, I write a blog, and we have, products that we recommend, but it's more information website than anything. I don't sell any products per se, but there are products that I test and recommend you'll find on there. A lot of people ask me about exogenous ketones, and the one that I recommend and prefer is Keto Start. There's a coffee creamer called Keto Brains that has like alpha GPC, MCT, lion's mane. That's a fantastic product. So these are 2 things I use every day. And then creatine, monohydrate, but I don't use a whole lot of supplements, maybe vitamin D3, omega-3s, but I eat a ton of fish, so probably don't need it. But yeah, ketonutrition.org, and sign up for the newsletter. I put a lot of little tidbits in there that I don't necessarily put on the site that I think could be helpful for people who want to learn more about this.
All right, Dom, thank you very much. Keep up the great work, and we'll look forward to more from you. Thanks, Dom. Take care. Have a great day. Maybe next time in Florida, I may chase you down. We'll get a workout and a steak or something. So anyway.
Absolutely. Yeah. Come to I would love to host you at the house. You can feel free to stay with us, and we got cows, we got weights, we got steak. So go fishing, go shooting, whatever you want to do.
Everything you need in life. Yeah. All right. Thanks, guys. Thanks, everybody. We'll see you back tomorrow. Thank you, Dom. Have a great day. Bye-bye, everybody.